So welcome back, everybody. We're picking back up from last week where we were discussing kind of the beginnings of the nervous system, how neurons are put together, and we're going to talk a little bit about those glial cells before we dive into our material for today. Okay, thanks, Alexis. Let me just get my Windows set up here. All right. So last time we had left off, we were talking about each of the kinds of different supporting cells that our nervous system has. Because remember, nervous tissue is fairly energetically costly to maintain, so it requires a lot of extra supporting cells to help it out, especially in the nervous system. The peripheral nervous system can kind of rely on some of the surrounding tissue to help support it and protect it and insulate it and provide nutrients to it as necessary. But the central nervous system is a little bit more insulated, so it needs a lot more supporting tissue to help it out in a proprietary sense. So kind of first up, kind of a catch-all central nervous system glial cell was the astrocytes. Remember, these are kind of like the processing cells that help determine what gets into the central nervous system, circulatory system, which is what we'll end up talking about at the end of today, the vasculature of the ventricles that create things like cerebrospinal fluid. If you've ever heard of like a Spinal Tap before, not the movie, which was a great movie, by the way, if you guys haven't seen it and you're into kind of like rockumentaries or mock rockumentaries, it's really funny. But anyway, spinal taps are where we sample things like cerebrospinal fluid, and astrocytes kind of determine what gets in there from the rest of the cardiovascular circulatory system. We also have ependymal cells, which in part are responsible for maintaining the fluid balance of the cerebrospinal fluid and help actually produce and line the cavities of the ventricular system of the brain and the spinal cord. And then we've got microglia, which kind of act as like the basic immune cells of the central nervous system. They can fight off infections and pathogens and various other kinds of mutagens as well. And then we've got our oligodendrocytes, which are responsible for myelinating or insulating the neurons of the central nervous system. And then out in the periphery, we have our Schwann cells that are responsible for helping make sure that the myelin stays intact on our peripheral nervous system neurons. All right. So that was what we'd ended with last time. So now, really, what I wanted to cover briefly today was the understanding of the action potential. So the action potential is literally just a contextualized membrane potential. So we had talked about this with the muscular system. We understand that membrane potential in simplified terms is literally just concentration gradients. But action potentials not only focus on the concentration gradients of ions, namely sodium being in higher concentration outside, potassium being in higher concentration inside. And due to the bulk number of sodium ions, we have a net positive charge outside of the membrane and a net negative charge on the inside of the membrane, in part due to the fewer number of positively charged things, but we also have negatively charged proteins that hang out inside of the membrane of our cell. In this case, our membrane is focused around the actual axon itself of our neuron. So understanding that membrane potentials really are just controlling and utilizing these different concentration gradients of ions, but also electricity. And action potentials are a perfect combination of both of those things working in concert with each other. This figure here is designed to show you how during a depolarization and then subsequent repolarization event of the membrane, the major protein that's responsible for helping repolarize the membrane, in other words, resetting it so that it can fire again, is this sodium potassium pump protein down here. So this orange guy. So this is an important one to take note of. This particular protein, every time it's activated or it churns through one of its cycles, it pumps out 3 sodium ions. So it shoves 3 sodium ions out into the extracellular space. In other words, moving them against their concentration gradient, because this particular protein, requires energy. So it's a form of active transport. And anytime we utilize active transport, we're generally pushing things against their concentration gradient. So it's kind of like pushing boulders up a hill so that we can just have them roll back down when we want them to. All right. And then, so every time we shove 3 sodium ions out, these little guys in pink, we bring in 2 potassium ions inside of the cell membrane. So that's every time this thing cycles through. All right. So this sodium potassium pump is important because it's the main protein that's responsible for maintaining these concentration gradients or these membrane potentials across these nerve cell membranes. Does it on nerve cell membranes, does it on muscle cell membranes, does it on heart muscle cell membranes, does it on intestinal epithelia? It does it everywhere in our body. We find this protein all over the place. Here in our neurons, it's even more important because it helps repolarize the membrane and lets the membrane reset so that it's ready for another signal to be able to stimulate it. So when we talk about an action potential, we typically will refer to this graph, right? So this one right here. We'll go back to the other figure in a second. This one actually should probably go here. So this figure graphs out what an action potential is. And the action potential is a measure of the change in millivolts across a membrane over time. So the way that we should think about millivolts is a form of potential energy, all right? Millivolts is a reflection of the charge that's built up due to the separation of these ions across the gradient. So it's a measure of potential energy. So if you're measuring the voltage of like a battery, or like if you're into music and work on guitars or anything like that, if you measure the voltage of certain pickups and you look at how the pickups kind of can have different kinds of voltages associated with them, that's reflective of if these circuits in a manner of speaking are closed. In other words, or sorry, not closed, we allow the current to pass through, we open up the circuit, then we're able to pass that current and make it active. So volts will change to current, measured usually in amps. So when we're talking about that, that's what we're referring to. So when you see this millivolts here, this is just a measure of potential energy, right? So these negative and positive numbers are also important to recognize because they're relative to the inside of the cell membrane, right? So the way that we measure this is we have a little electrode, or a voltmeter really, that has 2 electrodes. We have one end that's stuck outside the membrane, another one that's stuck inside the membrane. And so we're measuring is the difference in voltage across that membrane from the outside relative to the inside. So at rest, a nerve cell membrane sits at around minus 70 millivolts, okay? So it's a number that you should know and that you should memorize. And recognize that in order for a membrane to be depolarized, we have to reach something called a threshold potential. So this value in millivolts is generally around minus 55 millivolts to minus 60 millivolts, not positive 60, that'd be a lot. So I'll write those up here. So minus 55 millivolts to 60 or minus 60 millivolts is where our threshold potential sits, all right? So what that means and why that's important is our membranes have a little bit of wiggle room in what they'll respond to and what they won't respond to. So in order for an action potential to occur where we have complete depolarization of our nerve cell membrane, our axon, we have to get past that threshold potential. That doesn't happen all the time. We have a ton of what we call graded potentials that can fire at any point in time, and they're typically weak in their stimulus, and they can cause very small localized depolarization responses in the localized membrane. So why would that matter? Well, that matters if you're only trying to send a signal to a specific part of the membrane and you don't want it to propagate to the rest of the cell or move downstream on an axon. So this threshold potential kind of determines whether or not the membrane will respond in force or in mass to a stimulus, or if it can have a localized, graded stimulus response in the form of those graded potentials. So if a stimulus is strong enough to push a nerve cell's resting membrane potential past this threshold potential, we then undergo rapid depolarization. So keep in mind our units down here. for our x-axis. Our units are measured in milliseconds. Those milliseconds, again, this entire process happens in the span of about 1.5 to 2 milliseconds if you're talking about getting all the way back over here to the end of our figure, right? So 2/100ths of a second is how fast this happens, all right? And even if we're talking about specific parts of this, we're talking about fractions of that. So depolarization happens almost instantaneously, okay? So when our membranes depolarize during an action potential, they depolarize up to about 20 to sometimes 30 millivolts, positive 30 millivolts. That's important because what we care about is the magnitude of this difference right here, that depolarization, all right? So once the membrane has reached what we call like our maximal, our supramaximal depolarized potential, which is the absolute maximum limit that the membrane can be depolarized at, we then turn on and we activate these channel proteins that are receptive to sodium and potassium. Okay, what that allows for them to do is it allows for these ions to move passively back across the membrane, because during the depolarizing response, all of those ions move down their concentration gradients. So this will make more sense when we look at the next figure here in a minute, and we see how the gradients are changing. But then as that happens, we go through what's called repolarizing. It's essentially just resetting the nerve so that it can be ready for another stimulus, right? Think of it as like a catapult, right? You can't, I mean, you could technically have a catapult only go halfway, but what's the use in that, right? You want the catapult to completely launch that object as far as it'll go. That's the point of the catapult. And so in order to get that catapult reset again, we have to go through repolarizing. We got to wind it back down and get it ready for the next launch. So during this repolarizing phase, what we end up doing is we bring the membrane potential back down to below threshold. This is where our sodium potassium pump is starting to kick back in. And we end up going through this phase called hyperpolarization, where the membrane, resting membrane potential actually gets back below minus 70 millivolts. This is indicative of the sodium potassium pump being too good at what it does. So we end up creating too strong of a gradient. So then not only do we have channel proteins that let ions move down their gradients, we have these other kinds of channel proteins called leaky gated proteins. And just like most channel proteins, they're specific to a specific kind of ion. So we've got leaky sodium, leaky potassium, leaky calcium, leaky chlorine. We've got a bunch of different kinds, okay? So our sodium potassium pump is so good that we go through hyperpolarization before those leaky gated channels can catch up and say, whoa, 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 we've already got enough of these sodium ions down the gradient, or forced against the gradient. We can relieve some of the pressure here. So then we kind of turn on a little spigot, we let out some of that pressure, and we let those ions kind of get back to our resting membrane potential, which again, for human neurons is right around negative 70 millivolts, right? So I say that for humans because what's cool, amongst other vertebrates, cows, birds, fish, lizards, whatever, all of these animals can have different resting membrane potentials for their neurons. Even small mammals have, a little bit of physiological variance there. It's kind of cool. So this figure, this graph, summarizes a single action potential. If we're to equate this back to our muscle phys chapter from last time, you can equate a single action potential to a single muscle twitch response, right? So the greater the frequency of the nervous system stimulus of the muscle, then that translates to more frequent twitch contractions that, again, remember as last time, as we increase the frequency and we allow the muscle less and less time to relax, then the muscle will build up more and more tension as it's able to contract with more and more force, right? So now that we understand what our action potential graph is, what does this mean for the actual membrane of the neuron? Why do we care about this? Well, for one thing, we care about it because it's arguably the most commonplace and most prevalent physiological phenomenon that occurs in our body. They're happening all the time because our nervous system is always active. If it's not active, then you're brain dead, right? So First off, when our neurons are at resting potential, resting membrane potential, we're chilling at minus 70 millivolts. So all of our gated channels are closed, okay? So we've got all of our sodium outside and all of our potassium inside. Let's say we've got a stimulus that's strong enough to get us past that threshold potential to trigger depolarization, all right? So the stimulus can be a variety of different things. We have a bunch of different kinds of receptors that'll trigger an action potential. Some of the more common ones are things like chemical receptors that'll open if they see a particular chemical that they like. That's what we're going to talk about here in a minute. There's some that'll respond to electrical changes. We see those a lot in our graded potentials that can have some, little minor responses in that resting membrane potential. And then we have some mechanically gated channels that will open up if we physically deform them, if we change their shape that way. To put that in perspective, that's pretty much how most of our touch works, is we have a bunch of mechanoreceptors that are embedded within the membranes of those things like our Pacinian's corpuscle and our Meissner's corpuscles and our free nerve endings that we saw within the integumentary system. All of those will open up and cause a depolarization response if those membrane channels are opened physically. But during depolarization, our gated channels open up and we allow for those ions to flow down their concentration gradient. So as a result, we get a rapid depolarizing response. So the concentrations move down their own gradient, but more importantly, remember, just like we saw with the membrane potential in muscle cells where it was positive and negative and then it flipped, it's the same thing that happens here. That's what, by definition, what depolarization is. We flip the charge. The inside that was originally negative becomes positively charged because we've now let a bunch of sodium rush in. And then the outside that was originally positively charged ends up becoming negatively charged because we end up having more positively charged things inside the membrane, all right? So once that depolarizing response happens, then the membrane goes through repolarizing, where we close off things like our sodium channels, we end up pumping that out through the sodium-potassium pump, and then we allow for a little bit of potassium to kind of leak back out as necessary as we introduce that back in with the sodium-potassium pump too, all right? So this is one of those phenomenons that will pay off hugely if you understand it, because it'll explain how most everything else in our body operates when we talk about action potential propagation and the response in postsynaptic structures or organs or tissues or whatever it is. They're all going to be responding to action potentials here, okay? So before we move on, does anybody have any questions about the action potential? Or are we all equally confused right now? It's completely fine to admit that because this is a complicated thing to understand, and you shouldn't feel like, if you are solid on this, then awesome. That makes me really happy. But if you're confused about it, that's okay. Don't be ashamed because this is a tough thing to grasp. So if you have questions later, we can address those too. But if anybody has, a quick question that they want to ask now, we can answer that. Okay. Well, again, if you guys think of anything, or if you're too ashamed to, voice it, put it in the chat and I'll answer it as I can get to it. All right. So similarly to how electricity operates in like our houses and in our devices and things like that, our nerves conduct electricity. They're like electrical wires, and they can do it through two different ways of conduction. We have what's called continuous conduction, where the membrane itself will continuously propagate the change in the membrane potential downstream. So it'll move from left to right here. All right. So the membrane itself will just be sequentially depolarized downstream. It'll happen very quickly because this is, again, at the speed of electricity. And so we call this continuous depolarization. We typically find this process only occurring within unmyelinated neurons. So we find this a lot inside of our actual gray matter and white matter of our brain. We find it in little places within our spinal cord too, in these little guys called interneurons that typically don't have the myelin insulation that surrounds the axons. And again, that works because that protein and fat composition of myelin ends up blocking and obscuring continuous depolarization. So then how the heck is the membrane potential going to move downstream in a myelinated neuron? Well, they play hopscotch pretty much. So they end up moving downstream via a mechanism called saltatory propagation, which is where the charge, the depolarization of the membrane, will essentially hop in between each of the nodes of RON-VA as it moves downstream. So the action potential always moves downstream. And this might be something that we can, kind of look back, or we can come back to at the end of the lecture. And for those of you guys who are watching this later, think about that. Why does the action potential only move downstream? Why is it only going from left to right? Why isn't it going backwards? So think about that. So two major methods of signal conduction. We have continuous propagation through unmyelinated neurons, where the membrane is sequentially depolarized until it gets to its presynaptic terminal or telodendrite. And then we have one of the more common ways that occurs in myelinated neurons, where we have saltatory propagation, whereas we continuously depolarize these nodes of RON-VA, the signal will move downstream, and it's protected via that insulation, that Schwann cell of myelin, that helps make sure that the signal stays strong and intense so that it can reach each node of RON-VA, and then the signal can be depolarized again and again. That's the importance of the myelination, is we're able to maintain signal integrity and signal strength over long distances. Otherwise, our nerves would end up kind of petering out. The signals wouldn't reach where they're supposed to go. This is a really common occurrence in conditions like multiple sclerosis, where you have degradation of the myelin sheath that surrounds neurons, and then the neurons don't function the way that they're supposed to. All right. So now let's talk about the synapse. Okay. So this figure is important to understand and recognize, because this is where the action potential is going. So that action potential is just a signal, right? So it's just an electrical current. So that can change what it ends up doing to the target cell depending on what the end of the neuron contains. So in the ends of each of our neurons within these presynaptic terminals, aka telodendrites, okay, we have these little synaptic vesicles. They're little bubbles made out of membrane that hold chemicals. And these chemicals are what we call neurotransmitters. And neurotransmitters are what contain basically the nature of the signal that the neuron is sending out downstream. So these neurotransmitters, sometimes depending on where they're at, can be deemed neuromodulators, especially if they have a much greater, longer lasting effect on target tissues in the body. So when the action potential reaches this telodendrite, the first thing that happens is it depolarizes the membrane around here, and we open up calcium voltage gated channels. This is important because the calcium ions here act as what we call secondary signaling molecules. We're going to see this come up when we get into our endocrine system and the importance of secondary signaling molecules. Calcium ions are going to flood into the telodendrite here. All right. So what this does is this triggers a process called exocytosis. Exocytosis is basically where cells vomit things, all right? They shoot a bunch of stuff out of the inside of themselves, right? Endocytosis is when they eat, okay, and they introduce things back through their membrane. So through exocytosis, all of these little vesicles that contain our neurotransmitter are then going to migrate towards the end of the cell membrane right here and spew out all of those neurotransmitters into this space called the synaptic cleft, all right? So Dane, I'm looking at you. This is our little space. This is our gap that we were talking about last time with the neuromuscular junction. It's the same idea here. This occurs everywhere in our body. Anytime we have the nervous system interacting with another organ, we're always going to have a synaptic junction right there. So that's probably the better way to think about them. Neuromuscular is just specific to nerves and muscles. But anyway, those neurotransmitters will then cross this synaptic cleft. They do so really easily because it's a very small space and they can just move via diffusion. But then they end up attaching to what we call chemically gated ion channels on the other side of the cell membrane. So sometimes called chemically gated, other times called ligand gated, or I usually say ligand. Pretty sure it's pronounced ligand, but So chemically slash ligand channels. These are important because they don't open unless that chemical is present, or we have another chemical that can mimic that, all right, which is how basic pharmaceuticals work. This is how things like acetaminophen or ibuprofen work, is they basically can inhibit specific pain receptors that we have in our body called nociceptors, and they essentially deactivate our pain response, our pain stimulus, right? And that works really well if you have other kinds of opiates, things like morphine or opium, right? Those would be examples of what we call neuromodulators. So anyway, getting back here, with our neurotransmitters that get released here, and again, making that distinction, neurotransmitters are different than neuromodulators because neurotransmitters get broken down very, very quickly. Neuromodulators do not. They stay attached and they keep propagating that action potential, right? So here we've got our neurotransmitter that gets released into the synaptic cleft. It then finds the appropriate chemically gated channel on the postsynaptic membrane over here. Once that happens, we then open up these channels and let a specific kind of ion in. For our purposes, we're going to focus mostly on what we call a sodium gated channel. This is important because sometimes the gated channels aren't sodium, they're actually potassium, and that triggers a different response in the postsynaptic structure. Usually, if it's sodium, that's what we call an excitatory postsynaptic potential, and that will then keep the action potential going and propagate the signal and accentuate the effects. If a potassium gated channel gets activated by that particular receptor, then it usually dampens or negates the effects of the action potential. So why would this matter? Well, a quick example of that would be something like acetylcholine, which is the example that's talked about in the book and talked about with our muscle chapter. Acetylcholine is one of our most well understood neurotransmitters, and that one has excitatory effects on skeletal muscle, but has inhibitory effects if that neurotransmitter is released on cardiac muscle. So during bradycardia, or if we lower or slow down the heart rate, then we have a release of acetylcholine that then affects the cardiac muscle cells and then will dampen the action potentials that the heart muscle is receiving. Versus skeletal muscle, if we release acetylcholine on that, then that has an excitatory effect and the muscle will work more actively. All right. So yeah, this next table here has a couple examples of neurotransmitters. Serotonin and dopamine are mislabeled here. Those should be what we call neuromodulators, right? So they act on a lot more prolonged stage. So I won't go through all of these specifically, but you should check them out. One of the most common and well understood neurotransmitters that we have in our body is acetylcholine. We have a lot of acetylcholine receptors that occur all throughout our body, so it's very, very common. We also have epinephrine and norepinephrine receptors, again, other neurotransmitters that can accentuate or block or negate the effects of one another. So cocaine and amphetamines increase the release and block the reuptake of norepinephrine, resulting in overstimulation of postsynaptic neurons. So again, adding to kind of like a positive feedback loop of those things. In other words, that's why it's deemed excitatory. So again, you should take a look at those. It's pretty interesting to kind of look at where these things get released, what effects they have on the body, and some of those cool clinical examples. All right. Everybody with me so far? Give me a thumbs up if you guys are with me so far. All right, cool. Thanks, guys. I know it's a lot, but that's, I mean, this stuff is really fascinating to learn about. It's just a lot, right? So don't try and cram it all in your heads all at one time, like we're doing right now. But take this, just kind of chunk it out. I would say as you're going along in your notes, if you're taking notes as we're going along here, the main things I would focus on would be big ideas and main concepts. Don't worry about writing down every single little thing, but just kind of getting those big concepts and then going back later and fleshing those out on your own. So here we have an example of a neural pathway. So this is very, very similar to what we had talked about way back in chapter 1 when we were talking about how we have a input integration, and then output pathways. We've got three major parts to our homeostatic response. The nervous system operates in pretty much the same way because the nervous system is one of the main homeostatic regulatory systems. It controls pretty much everything. So what's important to recognize about this figure here is that with our sensory part of the circuit, you can think of it as kind of like a circuit here. Our sensory information is always going to be going back towards the spinal cord. And then all of our motor responses are the effector part of that signal chain, whereas sensory is the affecting part. The effector circuits always are going to be leaving and going away from the spinal cord. All right, so why does that matter? Well, if we were to do a cross section through the spinal cord, we're going to look at this in a little bit more detail here in a sec. We can see that we'll actually be able to determine where different signals are entering and or exiting the spinal cord. It's pretty cool. This right here is an example of a reflex arc where we can have this sensory neuron response here. It's not depicted, but there would be another branch. This would be, this sensory branch would actually have a diverging pathway that would end up having a little synapse onto one of the sensory nuclei of the posterior gray horn back here, and then send that information back up to the brain. So the brain will get the response and determine if we need to add to the effect of our reflex here. But this is showing us that we're actually kind of short-circuiting the brain and saying, okay, we're just going to hop back on our sensor, our interneuron right here, and then go back along this motor neuron to trigger one of our muscles here as a response to that. So that's our basic reflex arc, but we're going to go back and flesh that out here in a little bit. Typically in our nervous system, we have two different kinds of pathways. We have our converging and diverging pathways. So we see these all over the place. Typically with sensory neurons, we're always going to have diverging pathways because even if we have a reflex arc that occurs in our body, whether it's a withdrawal reflex, whether it's a pain reflex or like a pain withdrawal response, pretty much anything, the brain is always going to get the information later, right? If it's not instantaneous, it's going to be later, right? Because sometimes, even if the brain is going to get the information, it's a little bit too slow because sometimes we need to have a response so that we don't continue to injure ourselves. So if you're walking in the middle of the night and your little brother, your kid, your niece or nephew, whoever's staying over, they've got their Legos that are sprawled all over the floor, you step on that Lego, it's going to really suck if your body is like, well, I got to send that information back to the brain and then the brain has to decide what I want to do with it. Whereas, the way that our bodies normally work is we've got that immediate withdrawal response because holy cow, stepping on Legos in your bare feet is one of the worst pains in the entire world, right? So the point is with our sensory pathways, we typically have lots of diverging pathways. We have one that's going to end up going back up to the brain to integrate that information and say, okay, do we need to keep standing on one foot or is the pain away? And then we've got our other split pathway that's going to synapse onto that interneuron, and then that's going to go out and trigger the immediate motor response because, hey, that hurts, let's stop making that hurt. And then we have our converging pathways where we can have the integration of our higher orders of our central nervous system that say, crud, that really hurts. Now I'm going to start hopping on one foot because I'm holding my other one and I need to maintain my balance, right? So we've got our nerves that are going to be sending down more signals to the motor neurons that are going to be accentuating the effects of that motor response. So each part of our nervous system, both peripheral and central, are going to be working in concert with one another. Typically with the sensory nervous system, we have these diverging pathways. Within the central nervous system, we've got a lot of converging pathways that are going to be adding to the effects of things. All right. So we've got our breakdown of our spinal cord here. We've got a couple major bulges that actually occur throughout the spinal cord. We have a cervical bulge. We have the beginning of kind of like a thoracic bulge that occurs kind of in between our shoulder blades, which is indicative of what we call a plexus or a collection of well, not really nerve cell bodies, but nerve axons rather. And then we have a lumbosacral bulge, which is indicative of what we call the lumbosacral plexus. So those aren't depicted here, but those are found on the spinal cord. And so again, as we're seeing here with our peripheral nervous system, we have all these little yellow fibers that are exiting out of the spinal cord. and technically entering, as we'll figure out here in a little bit. The spinal cord itself actually ends at around the fourth lumbar vertebrae. So it ends way up here. Okay. And then we have an extension of that we call the cauda equina, because the first person to dissect this out thought that it looked like the tail of a horse. So cauda referring to the tailbone, equina referring to equine or horse. I think it was some Italian guy, which probably checks out. But so yeah, so those are kind of some of the basic extensions of the spinal cord that we see. All right, this here is a cross section through a traditional human spinal cord. This is right around the thoracic region, and we know that because of the presence of something called the lateral horn of the gray matter. This is going to contain sensory nuclei, which are basically little collections of nerve cell bodies that are only going to give information from organs or viscera found at the thoracic level. So that's why if you take a cross section through anywhere other than the thoracic vertebrae, you actually won't see the lateral gray horn there. But that's kind of a semantic thing. So the spinal cord itself is broken up into kind of two major areas. We have gray matter, and then we've got white matter. All right. So the gray matter, also called the gray, it's got, posterior and anterior gray horns that are associated with it, and lateral if you're in the thoracic region. Each of these are going to contain nerve cell bodies that are going to be either sensory or motor, right? So we have two major ones in the posterior and then two major ones in the anterior. And those are going to determine where sensory and motor signals are either coming or are going to, right? And then with our white matter, that's all out here, this is going to have a couple different columns that are associated with it. Each of these columns is going to contain different kinds of either sensory or motor information as well. That's passing all up and down the spinal cord. You can think of the spinal cord as like a big information highway. It's like it's like the 405 freeway. You got a bunch of lanes going one way, you got a bunch of lanes going the other way. And we've got all these little exits and off ramps where signals can either leave the spinal cord or enter the spinal cord. So that's another like kind of an acronym, or not acronym is not the right word, but I don't know. Analogy, that's the word I'm looking for. It's another analogy I like to use to describe the nervous system, is it's really very similar to like a freeway system, where each of our cars you can think of as, one kind of information signal that's going one way, and you can have all these little interchanges and you can switch freeways. Oh crud, I forgot my exit. So you can exit and then go back on and hop on, another on ramp, and then, take your correct exit if you need to. Or if there's construction, or if, part of the freeway is closed, then there's other multiple ways for information and signals to get to where they need to go. All right. Okay, so some of the other anatomical parts of our spinal cord here are these kind of spaghetti looking things right here, which are what we call rootlets. So each of these rootlets is going to contain multiple nerve fibers that are either entering the spinal cord, as we would see with our dorsal root, or exiting the spinal cord, as we would see in our ventral root. So if you're looking at the dorsal root of the spinal cord, the dorsal root is always carrying sensory neurons. Okay. And then the ventral root is always going to be carrying motor neurons. Okay. So where it gets a little bit annoying, though, is as we get farther away from the spinal cord here, the connective tissue, called the epineurium, ends up housing both motor and sensory nerve fibers. So what does that look like in reality? Well, if you like chicken and you like eating chicken wings, oftentimes if you eat chicken wings, like you might get a little bit of gristle next to the bone. It's usually nerves. And that nerve that you're seeing there is actually containing both the sensory and motor neuron information that's going to that part of the body. Usually in a chicken wing, it's the musculocutaneous nerve. But that's what we're seeing. If we see a nerve that's outside or in the peripheral nervous system, is each of those nerves are going to contain both sensory and motor neuron information because it's all wrapped up in the same connective tissue. But remember and keep in mind that the actual nerve fibers are going to be completely separate from one another. They're insulated, they're myelinated, they're all separate, but they're just wrapped up in the same connective tissue. All right. Okay. Everybody with me so far? Hopefully. Yep. Okay, cool. So here's another example of converging and diverging pathways. So we see with our sensory root in green here, going back into our spinal cord via the dorsal root. They enter via the dorsal rootlets, and they can synapse onto various sensory nuclei in the posterior gray horn back here, and that signal can diverge and then end up in some of those lateral and posterior columns that can contain specific neurons that are going to send that information back to the brain. And depending on whatever the issue is, we can accentuate or add to the effect of a reflex, or whatever. And then we've got our individual motor neurons that are going to be exiting via the ventral root down here in the ventral rootlets and the ventral motor nuclei of the anterior gray horn. We call this the anterior side because this is the side that's closest to the belly. This side back here is called the posterior or dorsal area because that's going to be closest to kind of our spinous process. So dorsal and lateral and ventral white columns can contain various kinds of sensory neurons that will take that information back up to the brain and then to different regions to decide what it wants to do with that information. And then we've got examples of our interneurons here, but then several other neurons that can contain converging signals that are being brought back down via higher centers of the brain. All right. Okay. So here we've got another infographic showing that as well. So we've got our converging pathway here. So we've got our motor nuclei that's going to be it's going to be carrying information back down from the brain down to affect the skeletal muscle. And we've got our diverging pathway where we have those sensory neurons that can synapse onto various white matter columns that are taking that information back up to the brain.
Can I ask a question?
Yes, of course.
Okay, because I think I'm a little confused. I could be, but I'm not sure. So just as a general example, let's just say I touch a stove with my finger.
Mm-hmm.
That's the entry sensory, I guess, receptor kind of thing. It sends it up to my brain and it figures out where it's going to send it, comes right back down that same channel, and then it spurs it out to wherever it needs to, wherever the pain's going to be felt or things like that. Is that like the general idea?
Yeah, sort of. So the same channel thing is not strictly correct. So what we'll find out is we have different essentially it would be kind of like, so like once you get like, so do you take the freeway to your job, Pamela? Okay, so it'd be like, so if I don't know which direction you drive, but let's say that you take the eastbound freeway, right, to get to your job. Let's say let's say you live in Riverside and you work out in Indio, right? You work at Coachella and you have to drive out to Coachella every day. It'd be like once you got there, it would be like taking the same side of the freeway, but back to your home, right? You can't do that. Otherwise you get into a big traffic.
So it's like it's like one channel but two different directions kind of thing, or
Yeah,
two different channels just like kind of stuck together, one going one way, the other one going kind of like this.
Yes, that'd be a
Okay.
good way of thinking about it. Yeah.
Okay.
Absolutely.
Okay. Thank you.
Cool. Yeah. Thanks for your question. Yeah. So that's generally what's going on here. Is anytime we have sensory input, we have one channel, one neural pathway, and then anytime we have motor output, we have a different neural pathway. They can travel on the same freeway, but just on different sides of it, if that's kind of what you're asking. Okay, so but in that same vein, let's talk a little bit about some reflex arcs. So we've got a couple different examples of reflex arcs. We have this one, which is what's called a stretch reflex. These are pretty cool. Inside of our skeletal muscles, we have these specialized kinds of motor neuron receptors. that are called gamma receptors. And these guys are responsible for recognizing if the muscle gets stretched too far, if it gets pulled beyond its normal operating length too much. So we have this cool withdrawal response that says, hey, we need to make sure that we don't damage this tissue that much. So we're going to trigger a very small twitch contraction to reintroduce or bring the muscle back to its original resting length. A great example of this is if you go to the doctor and you get the hammer tap test on your knee, where we tap the patellar ligament down here. What that ends up doing is stretching our patellar tendon up here that then pulls on the quadriceps muscles. When that ends up happening, it stretches those muscles very quickly past that normal operating length. So the trigger here to recognize is that if we stretch the muscles too quickly, not if we just gradually stretch them and stretch them and stretch them. If we stretch it too quickly, then the body's like, ooh, that we don't like that. We want to make sure that we, don't damage our muscles too badly. So we have sensory neurons that are gamma sensory neurons and gamma motor neurons that end up recognizing that and feed back into themselves and create a stretch withdrawal response. So these muscles here will contract minimally, small twitch contraction, to pull the muscles back into their normal resting length. And then as a result, we end up kicking our leg out too because of where the muscles are attached. This is an example of what we call a monosynaptic reflex because we only use one synapse. That's what we're seeing here. So our little single synapse right here onto this motor neuron, boom. So these gamma motor neurons are not capable of introducing very strong electrical responses and depolarizing responses. Okay, they're very minimal. All right. So monosynaptic reflexes. But then it gets really cool when we start looking at things like withdrawal or pain reflexes. This is typically where we have polysynaptic reflex arcs that come into play. Because, so like, for example, Pamela, using your example of like, if you grab like a hot cast iron pan and you forgot that you were letting the pan heat up for a while because cast iron's dumb and you have to let it heat up, but cast iron's also really cool because you can do a lot of stuff with it. But anyway, let's say you forget you're heating up your cast iron and you forget, oh crud, it's been sitting on the stove for 10 minutes now. This thing's really hot. So you grab it, and then the second that you touch it, your brain's like, whoa, that's really hot. What ends up happening is we send that sensory information back to our spinal cord, and then the information is going to diverge and go back up to our brain. While that signal is going back up to our brain, our body understands that like, hey, this is really hot. We don't want to have to sit here and let our brain decide if we want to let go of it or not, because that's going to take a lot of time. So we're going to synapse onto interneurons that are going to then activate the appropriate alpha motor neurons, which are the really strong motor neurons that can trigger a tetanic contraction and pull our arm away with a strong response. So polysynaptic neurons are really interesting because they can get fairly complex. What we want to recognize is that, in this example here, this is a very odd example that your book has. I'm not the biggest fan of it because of the way that it's being shown here. This pain stimulus would be a very weird one. It'd be like if you're sitting down and somebody stuck a tack into your heel. It's not very realistic unless, you happen to know a prankster or something like that. But yeah, so like if this were to occur, the sensory response here would be to activate the flexor muscles to withdraw the limb away from the stimulus. That's pretty self-explanatory. But more realistically is like if we grab a hot pan or if we step on something that we don't like, then what ends up happening is we not only have to make sure that we withdraw our limb away from that particular stimulus, we got to make sure that we're only activating the right kinds of muscles. Because as we'll talk about in lab today, we have what are called flexor muscles and we have what are called extensor muscles, right? So with our arm example for what Pamela was talking about with grabbing the pan, we want to make sure that when we grab that pan that we don't activate our triceps because our triceps will push our arm into that stimulus. We want to make sure that we only activate our biceps muscles, which flex our arm and bring that stimulus away. And we want to deactivate our extensors. So polysynaptic pain withdrawal responses will usually involve activating flexor muscles while deactivating extensor muscles. Okay, that's usually what ends up happening. And then it gets even more intricate when we talk about the Lego example, because then that signal will actually cross our little isthmus of our gray matter horn right here. And then that will synapse onto multiple motor neurons that'll inhibit flexors and activate extensors on the opposite side of the body. So that when we, shift our weight to our leg doesn't just immediately collapse as a result. So we can get really complicated, cool polysynaptic reflexes. And then it gets even weirder when you talk about reflexes in response to hearing or sight or, photopupillary reflexes, all that kind of cool stuff. And then I've got a little quick blurb here on spinal cord injuries. I'll summarize this because we're running low on time. Long story short, spinal cord injuries are variable and they're usually different, across every single patient. No one spinal cord injury is generally alike. You can have the same area that's injured, but depending on the nature of the injury, the spinal cord can react very differently. Generally, the best thing to do is to try and immobilize the area and get anti-inflammatory drugs as fast as possible. So we have methylprednisone as a really common anti-inflammatory drug that will reduce the swelling surrounding the spinal cord. The reason you want to reduce the swelling is so that we can allow for medication to reduce damage to the tissue, and then the tissue can actually heal and repair itself effectively. If it becomes too swollen, then you have ischemia that'll occur, then the tissue will senesce and die. And nerves don't really do a great job of repairing themselves once they're dead. Usually once they're gone, they're gone. You can reverse a lot of that damage with things like physical therapy or anything like that, but it takes a lot of work to do so. All right. All righty. Really quickly, we have something called the dermatome map. If any of you guys are into acupuncture or are familiar with that, this is what acupuncturists study and utilize to great effect. With different kinds of stimuli, they can trigger specific neurons that will cause different responses throughout the body. It's really cool. I don't know a ton about it, but the dermatome map is heavily studied in acupuncture school because the way that we're developed in layers and sections, and all of our skin and organs and everything is linked to every area of the spine and where the nerves exit the spine. So when we're talking about acupuncture, we're stimulating different neurons and causing different kinds of action potentials to be propagated throughout the body. It's pretty cool. So we've got all of these different nerves. They're all linked up to different areas of the body. So you can use an understanding of the dermatome map to test for things like spinal cord damage. There's a lot of really cool stuff that you can do with it. All right. Let's move on here. So we've got various kinds of plexuses where spinal nerves exit the spine. So as I mentioned, we have our cervical plexus up in the neck. We have our brachial plexus that's in our armpit. And then we have our lumbosacral plexus that's down in our back. So lots of these are going to be the main nerves that you're responsible for understanding what muscles they innervate and what areas of the skin they innervate as well. So sensory and motor information. So this is important to recognize too. All right. Okay, let's talk a little bit about the brain. So here's a cross section through our brain. Not everything is labeled, but a lot of stuff is labeled. We have our large portion of our brain making up most of the gray matter of our brain, called the cerebrum up here. The cerebrum has multiple folds in it that are important for storing memory, for reflex responses, for integrating different kinds of stimuli. All of that happens up here in the cerebrum and the corpus callosum, in the diencephalon, where we have things like our thalamus and our hypothalamus. Back here in the cerebellum, things that integrate, repetitive movements. This big old organ right here does a lot of really, really cool things. Okay. So we've got our cerebrum up here, posterior, anterior side. We have our pre-midbrain, what we call the diencephalon, which contains things like the thymus and the hypothalamus. Thalamus and the hypothalamus. And then we've got our midbrain just here, the pons, and then the medulla oblongata just down here. All right, so let's talk a little bit about what those do. Cerebrum, biggest part of the brain, controls higher mental function, divided into left and right parts. Surface layer of the gray matter that we call the neural cortex. So it's like a coating and protective material that covers the cerebrum. Folded surface area or folded surface increases surface area. So again, kind of cheating the laws of physics. We can cram a lot more information into places where we have all these folds. So we have these elevated ridges called gyri, shallow depressions called sulci, or an individual one being a sulcus, and then deep grooves called fissures. And the development of these fissures and gyri and sulci are important for making sure that you have normal brain development. So I've got labeled on here. We have kind of one of our folds called a gyri, and into a little indent called a sulci right here. So this would be an example of a sulci, sulci, sulci right there, a little sulci. But anytime you see like a really dark indentation, that's usually a fissure, right? And so all of these things have names. All of these fissures, sulci, gyri have names. Each of them does something specifically, all right? And you have to know all of them. I'm just kidding. Definitely don't. That would be ridiculous. We have the cerebellum, second largest part of the brain, has two major hemispheres, coordinates repetitive body movements, and covered with its own kind of cortex called the cerebellar cortex. And then we've got the diencephalon down here, located underneath our limbic system, where we have the corpus callosum, just here. So links the cerebrum with the brainstem. Okay, we have a left and right thalamus, and then our hypothalamus. Each of the thalamus and the hypothalamus have different portions to them, different kinds of nuclei that control different kinds of reflexes, sensory integration, different kinds of neurons that house different neurotransmitters that can contain different hormones that can be released to various parts of the body as well. So hypothalamus, hormone production, regulates things like emotion, controls various aspects of autonomic function. Then we have our pituitary gland, major endocrine gland connected to the hypothalamus via a little funny word called the infundibulum. That's how you pronounce that. And it interfaces with the nervous and the endocrine systems of the body. And then we get down here into the brainstem. All right. So the brainstem includes the mesencephalon. Some people pronounce it mesencephalon. You can call it mesencephalon. You can call it stupid word, whatever. I don't know. Whatever you guys want to call it on your own. That's totally fine. Mesencephalon, pons, and then medulla oblongata. Mesencephalon processes sight, sound, and associated reflexes like withdrawal responses to sight and sound, or it can help integrate our fight or flight response in response to loud sounds or bright lights or anything like that. It's also responsible for, it's one of the main sites for integration of memory and reflex, one of the major sites for treatment for individuals with post-traumatic stress disorder in combination with the thalamus and the hypothalamus. Each of those areas can contribute to the brain placing yourself in a specific scenario given a particular kind of stimuli. Then we have the pons, connected to the cerebellum, or connects the cerebellum to the brainstem, is involved in somatic and visceral motor control. So helps regulate things like peristaltic motion, so movement of food through the intestines, but also helping control or regulate different kinds of skeletal muscle activity. And then the medulla oblongata relays information to the brain from the spinal cord. This is where we actually have information from the left and right side of our body cross over to the left and right parts of our cerebrum and our cerebellum and other aspects of our brain. Regulates autonomic functions like heart rate, blood pressure, and actual digestion or the physical breakdown of our food. Okay. So then we get over here to the diencephalon. So here we've got our thalamus right here. And then we have the interthalamic adhesion. So we've got two little lobes of the thalamus. And each of these little nuclei are going to contain different neurons that respond to different kinds of stimuli. And then we've got our hypothalamus, which has all of its own nuclei that is then connected down here to the pituitary gland. So if you guys remember when we were talking about the skull and when you talked about that structure, the sella turcica, this is the little guy that's hanging out in the sella turcica. It's a really, really important gland for the endocrine system. We're going to come back to that as well. And then we've got the pineal gland, which is kind of on the back part of the limbic system right here. This guy produces melatonin, which helps regulate our sleep cycle and controls our circadian rhythm. Okay. All right. Let's see here. So yeah, now let's talk a little bit about some of those ascending and descending tracks that I had mentioned that are going to be making their way through the white and gray matter of the spinal cord. So we've got a couple different pathways. So we've got the spinothalamic, dorsal column, and spinocerebellar. We have anterior and posterior components to that. So spinothalamic being pain, temperature, light touch, pressure, tickle, and itch sensations. And then dorsal column contains proprioceptors, touch, deep pressure, and vibration. And then spinocerebellar, proprioception to the cerebellum. This is one of my favorite words to throw out there, is this word proprioception. It basically is what controls our brain's understanding of where our bodies exist in three-dimensional space without having to actually see your limb. So like, for example, I know my arm is behind me right now. I'm touching the top of my little mountain up here, okay? I know that that's back there. I don't have to look and see that it is. I know that it is because of the proprioceptors, or the proprioception, the sense of proprioception is how my brain knows where my body exists in three dimensions. So here we've got our dorsal columns, the posterior spinocerebellar tract, anterior spinothalamic tract, and anterior spinocerebellar tract just here. So here's where they sit in the white matter. So they're not part of the gray matter. These are all just part of the white matter. And so we've got these on the lateral, or on the, the right side. No, it would be the left side of the spinal cord over here. So this is our anterior aspect. So this would be left. I'm sorry, right and then left over here. And so the way that looks as it makes its way through the spinal cord is we then have synapses that cross over from the right side to the left side via the medulla oblongata. And then we've got our second neuron in the pathway that passes all the way up to the thalamus and then can integrate to whatever part of the cortex it needs to go to for somatic sensory. All right. And then we have a kind of generalized breakdown of different regions of the cerebrum and what they're responsible for. So we have our primary motor cortex right here in dark purple, premotor area, prefrontal area, motor speech area. So as I'm talking right now, all of that's controlled by what we call Broca's area right here, kind of part of the temporal lobe. Then we have our primary auditory cortex, auditory association. So being able to hear, understanding what you're hearing, and then sensory speech, being able to recognize what words you are forming and what words you want to form, okay? Sometimes affected by stroke, one of the areas that is affected by stroke and can limit people's ability to form coherent speech. And then we've got our visual association area, and then our visual cortex that houses the neurons that do the job of flipping the information or flipping the images that we're seeing right side up so that we can see them the way that they're supposed to. Somatic sensory, primary somatic sensory cortex up here. Okay. All right. So then moving down here to other parts of our white matter. So we've got our a couple different pathways of direct and indirect pathways of our descending tracks. So these would be all of our motor neuron tracks. We have lateral corticospinal and anterior. Muscle tone and skilled movements, especially of hands. So lateral corticospinal tract responsible for dexterity, for whatever your, kind of 360 no scope Call of Duty headshots are, right? And then we've got our muscle tone and movement of trunk muscles. So lots of our spinal extensors and spinal flexor muscles to utilize in this anterior corticospinal tract. And then we have indirect. Rubrospinal, reticulospinal, vestibulospinal, and tectospinal tract ways. So rubro uses movement coordination, or controls movement coordination. Reticulo uses posture adjustment, especially during movement. So kind of fine motor control as we're moving. So if you're walking, and if you're like me and you're really tall and you have to duck, reticulospinal pathway, posture adjustment. Vestibulospinal, again, accentuating things like posture and balance. And then tecto, movement in response to visual reflexes. All right. So here's where we find each of those pathways, each of those columns. So we have our lateral corticospinal and rubrospinal just here, right next to our other pathways. Let me see, I've already forgotten the names of those. We have our posterior spinocerebellar tracts just here. That's what they're called, spinocerebellar. just here, spinocerebellar tracts. So posterior and anterior. And then we've got our anterior corticospinal, and then the reticulo and the vestibulo and tectospinal tract pathways over here. Okay. Right. So again, motor cortex from the same area passes down the left side of the body, synapses, and transfers over in the medulla oblongata. So that's where we have crossover from the left to the right side of the body. All right, let's see. So this is kind of cool. Our brain can, this is what I was talking about with accentuating the effects of a response to a reflex. Sometimes the reflexes are super exaggerated and we don't need to have that same kind of a magnitude of response to a particular stimulus, right? So we have this cerebellar comparator function, which is where we can coordinate smooth and skeletal muscle information or activity with sensory information. So this is how we're able to control not only our responses to things, but how we can fine tune our muscle activity to what we want them to do. For example, when you pick up an egg, you're not going to use the same force that you would pick up a 30-pound dumbbell weight with, right? You would crush that egg. That's where our cerebellar comparator function comes into play. So again, we have our motor cortex, which sends out the action potential to lower motor neurons in the spinal cord. Action potentials from the motor cortex inform the cerebellum of the intended movement. Cool. Lower motor neurons in the spinal cord send action potentials to skeletal muscles, causing them to contract. Again, we have these cool proprioceptor neurons that then determine where our muscles are at in three-dimensional space. Proprioceptive signals from the skeletal muscles and joints to the cerebellum convey information concerning the status of the muscles and structure, being moved during the contraction, right? So we've got a cool little feedback loop here. The cerebellum then compares the information from the motor cortex with the proprioceptive information from the skeletal muscle joints. So as you go and you pick up that egg, and as you're starting to activate those muscle or those motor neurons, we're getting a feedback signal that's saying, all right, cool, this isn't super heavy. We don't have to activate the rest of our motor neurons to activate more muscle fibers, all right? So action potentials from the cerebellum to the spinal cord modify the stimulation from the motor cortex to the lower motor neurons. Action potentials from the cerebellum are sent to the motor cortex to modify its motor activity if necessary, all right? So again, different kinds of stimuli or different numbers of stimuli versus if we are picking up an egg or a 30-pound dumbbell. Or if you are blindfolded, it's the same idea. That's why you don't need to see something in order to understand how much force you need to apply to something. All of those proprioceptors that we have in our joints and in our muscles give our brain all the information that it needs to interact with our surroundings. All right. Okay, let's talk about brain waves with a little bit of time that we have left. All right. Brain waves. So we've got a couple different kinds of brain waves. Alpha waves are kind of normal awake brain waves that we have. We can start to elicit beta waves where we have intense mental activity, common when you see or for what you see at the beginning of sleep, or if you're trying to follow along with Mr. Beachco's lecture on the central nervous system, completely fine. Also, you might be experiencing theta waves if you're a little bit frustrated right now. If your brain is confused and doesn't like what it's hearing right now because it's just too much, that might happen. It's actually more common in people who have things like schizophrenia or bipolar mood disorder as well. And then we have delta waves. So this is where we have sleep in infants or traumatic brain injury in patients. We can have delta waves that are elicited here. I don't have enough time to talk about memory right now. It's really cool. You guys you're still responsible for the material in the textbook, but you should look over it. If you have questions, let me know and email me. Basically, the way the brain works with memory is that it physically changes as we encounter new memories and we store them in different ways. And we can access those memories differently through all these cool different kinds of memory methods. One of the best things to do, this is your shortcut for memorizing new things, and we do this a lot already, but using what are called memory engrams. So we can relate new information to old information that we're already well familiar with. So I like to throw out a lot of analogies about Lord of the Rings because I love Lord of the Rings. But I also, can form up a bunch of different analogies to other things that I like as well. It's the same idea. You can trick your brain into being able to consolidate information in a much more efficient way by tying new complicated concepts into things that you already understand. All right, limbic system. Okay, limbic system is pretty cool. It's centered inside, kind of inferior to the cerebrum. We've got a bunch of these different kinds of structures that make up the limbic system. Helps determine things like long-term memory, our emotional and visceral responses to different kinds of stimuli. So, that elation at getting engaged, or, finding out if you're having a kid or something like that. All of those emotions are controlled through things like the limbic system. It also controls things like our mood stability, regulates our motivation, ambition. All of those kinds of things are regulated through the limbic system. All right. Then let's see. We unfortunately don't have enough time to talk about the cranial nerves right now, so we'll wrap that up at the beginning of our lecture on Thursday. So let's see. The last thing that we'll discuss are the meninges, so the connective tissue that surrounds the brain, and then we'll look at the ventricular system of the brain too. So the meninges here are basically just 3 layers of connective tissue that the brain has that surrounds the spinal cord and the brain. So the outermost one, the superficial layer, is called the dura mater, which in German literally translates to tough mother. That's what it translates to. Then we have this subdural space, okay, that separates it from our next layer called the arachnoid mater. So the arachnoid mater then sits here directly underneath our dura mater. We have 2 different components to the dura mater. There's actually a fluid that occupies the subdural space, part of the ventricular system. And then that helps, that's in part maintained by the arachnoid mater tissue. It helps maintain some of that fluid concentration. And then deep to the arachnoid mater, we have the pia mater, which is our thinnest layer of connective tissue, houses things like blood vessels and veins, which help supply blood to the central nervous system tissue. And then we've got our limbic system here, which is basically a series of ventricles or open channels within our brain and our spinal cord that produce cerebrospinal fluid. Why do we need to do that? Well, it's one of the major ways that we're able to transport fluid and nutrients all over the central nervous system. So all the way down our spinal cord and all throughout our brain, we have these open channels that are fluid filled with cerebrospinal fluid, all right? So like I mentioned, we have this sagittal sinus, or the space between the what's called the outer, well, I forget what the name of it is. Is it the endosteal dura mater? It's basically the layer that sits underneath the cranial bone. And then we have the other layer that essentially grafts right up against the arachnoid mater. The arachnoid mater has these little granulation cells, which are protrusions that essentially elicit more and more cerebrospinal fluid. They help contribute to that concoction. So then our brain actually sits inside of this little fluid-filled case right here. So we've got the inside of our cerebrum that has a bunch of fluid via the lateral ventricles. And then we've got our third ventricle that produces cerebrospinal fluid via the choroid plexus, a little collection of cells. It's part of the arachnoid mater. And then we have this cerebral aqueduct that connects the third ventricle here down to the fourth ventricle, which links all of our cerebrospinal fluid from the brain down into the spinal cord. Okay. So here's, like I mentioned before, or at the beginning of the lecture, when we were talking about the ependymal cells, we see these guys all along the choroid plexus, and they help contribute to our cerebrospinal fluid production. All right, so we're going to end here for today, and we'll finish up our discussion on this chapter at the beginning of Thursday, where we talk about our cranial nerves.